Good day, ma'am. My name is Irish Faith Argalas and this is my report. So, my topic is all about recombinant DNA technology, and we have here the objectives. First, define recombinant DNA technology and its core purpose. Second is identify the key tools, enzymes, vectors, and hosts, and the outline of the five main steps of the process. Lastly, the applications. Give at least three real-world applications of the technology in medicine and agriculture. A technique mainly used to change the phenotype of an organism host when a genetically altered vector is introduced and integrated into the genome of the organism. So basically, this process involves the foreign introduction of piece of DNA structure into the genome which contains our genes of interest. This gene which is introduced is the recombinant gene, and the technique is called the recombinant DNA technology. There are multiple steps, tools, and the other specific procedures followed in the recombinant DNA technology, which is used for producing artificial DNA to generate the desired product. Let's understand each steps of in detail. First is what is recombinant DNA technology. The technology used for producing artificial DNA, though the combination of different genetic materials from different sources, is referred to as recombinant DNA technology. Recombinant DNA technology is popularly known as genetic engineering. The recombinant DNA technology emerged with the discovery of restriction enzymes in the year of 1968 by Swiss microbiologist Werner Arber. Inserting the desired gene in the genome of the host is not as easy as it sounds. It involves the selection of the desired gene for administrations into the host, followed by the selection of the perfect vector with which the gene has to be integrated and recombinant DNA form. Thus, the recombinant DNA has to be introduced in the host, and at last, it has to be maintained in the host and carried forward to the offspring. Next is tools of recombinant DNA technology. The enzymes, which include the restriction enzymes, helps to cut. The polymerases helps to synthesize, and the ligases help to bend. The restriction enzymes used in recombinant DNA technology play a major role in determining the location at which the desired gene is inserted into the vector genome. They are two types, namely endonucleases and exonucleases. The endonucleases cut within the DNA strands, whereas the exonucleases remove the nucleotides from the ends of that strand. The restriction endonucleases are sequence-specific, which are usually palindrome sequences, and cut the DNA at specific points. They scrutinize the length of DNA and make the cut at a specific site called the restriction site. Next is the vectors. It helps in carrying and integrating the desired gene. These forms a very important part of the tools of recombinant DNA technology, as they are the ultimately vehicles that carry forward the desired gene into the host organism. Plasmid and bacteriophages are the most common vectors in the recombinant DNA that are used, as they have a very high copy number. The vectors are made up of origin of replication. This is the sequence of nucleotides from where the replication starts. A selectable marker constitute genes which shows resistance to certain antibiotics like ampicillin and cloning sites. The sites recognized by the restriction enzymes where desired DNAs are inserted. Host organism, into which the recombinant DNA is introduced. The host is the ultimate tool of recombinant DNA technology, which takes in the vector engineered with the desired DNA with the help of the enzymes. There are a number of ways in which these recombinant DNAs are inserted into the host, namely microinjection, ballistic or gene gun, alternate cooling, and heating. Process of recombinant technology DNA. The complete process of recombinant DNA technology includes multiple steps maintained in a specific sequence to generate the desired product. First is step 1, isolation of genetic material. The first of the initial step is in recombinant DNA technology is isolate the desired DNA in its pure form, i.e. from or free from other micromolecules. Step 2, cutting the genes at the recognition sites. The restriction enzymes play a major role in determining the location at which the desired gene is inserted into the vector genome. These reactions are called restriction enzyme digestion. Step 3. Amplifying the gene copies through polymerase chain reaction or PCR. It is a process to amplify a single copy of DNA into thousands of millions of copies once the proper gene of interest has been cut using restriction enzymes. Step 4. Ligation of DNA molecules. In this step of ligation, the joining of two pieces, a cut fragment of DNA and the vector together with the help of the enzyme DNA ligase. Step 5. Lastly, insertion of recombinant DNA into the host. In this step, the recombinant DNA is introduced a recipient host cell. This process is termed as transformation. Once the recombinant DNA is inserted into the host cell. Lastly, the application of recombinant DNA technology. DNA technology is also used as to detect the presence of HIV in the person. Gene therapy, it is the use of the attempt to correct the gene defects which give rise to hereditary diseases. Clinical diagnosis or ELISA is an example where the application of recombinant. Recombinant DNA technology is widely used in agriculture to produce genetically modified organisms such as flavor saver tomatoes, golden rice, rich in proteins, and BT cotton to protect the plant against bollworms and a lot more. In the fields of medicine, recombinant DNA technology is used for the production of insulin. Recombinant DNA technology or genetic engineering let us make artificial DNA by combining genes from different sources to change how organism look or works. It started with the discovery of restriction enzymes in 1968 and uses tools like enzymes to cut and join DNA, vectors to carry genes, and host organisms to grow to the new DNA. The process of 5 main steps get the desired DNA, cut it more like copies, join it to the vector, and put it in the host. So that's all for my report. My name is Irish Faith Argalas from Best in Science 2, representing the recombinant DNA technology.